Hello everybody, here is Medmora. If you are interested in medical subjects, don't forget to subscribe my channel. In this video, we'll learn the action potential or the depolarization and repolarization of cardiac myocytes. In this figure, we can see the electrical conduction system of the heart. We have two separate action potentials in heart, pacemaker potential in electrical conduction system and action potential in cardiomyocytes, which is different from the pacemaker potential. In this video, we will learn action potential in cardiomyocytes. First, we should note that in any cells in our body, there is a difference in charge from inside the cell compared to outside the cell. The charge inside the cell compared to the charge outside the cell is negative. Now, what is the reasons of this difference? There are two major reasons for this difference. The first reason is that all the cells in our body have a pump in their cell membrane. This pump's function is to take free sodium from the inside of the cell and pumps them to the outside of the cell. And at the same time, it takes 2 potassium from the outside of the cell and pumps them in. So by throwing 3 positive sodium out but bringing 2 positive potassium back in, we will have a net positive charge outside the cell compared to inside the cell. This pump is called sodium-potassium ATPase pump. This sodium-potassium ATPase pump only caused the difference negative 5 mV. However, the difference in cardiomyocytes is negative 90 mV. So what causes the other negative 85 mV? In all the cells in our body, there is a high concentration of sodium outside the cell and a high concentration of potassium inside the cell. So sodium wants to diffuse in and potassium wants to diffuse out. This diffusion is by specific channels, sodium-specific channels and potassium-specific channels. In resting situation, when cardiomyocyte is not contracting, these sodium channels are closed, so sodium cannot enter the cell. But some potassium channels are still open and let some potassium to diffuse out. Letting potassium to leave the cell makes inside more negative compared to outside, so it makes other negative 85 mV. So now we understand the reason of this part of the figure. Let's understand why this part of the figure is like this. When sodium channels open, sodium rushes into the cell. So now inside is positive compared to outside, and it reaches positive 20 mV. Going from being negative 90 mV inside the cell to being positive 20 mV inside the cell is called depolarization or phase 0. In phase 1 or initial repolarization, by reaching positive 20 mV, all the sodium channels close and potassium channels start to open up. So potassium starts to diffuse out because it is positive while leaving the cell make inside more negative. And this part of the figure will happen. There is also another type of channels in cardiomyocytes membrane. L-type calcium channels. It open up and close slowly, and it is also source of calcium for contraction of cardiac muscles. After initial repolarization, calcium channels open and potassium channels are still open. So potassium comes out and calcium comes in. Both of them are positive, so they are in balance with each other, and the potential of membrane will be stable. This phase is called plateau. Plateau is shown in the figure. In phase 3, calcium channels close, but the potassium channels remain open. So potassium leave the cell and make inside more negative and repolarization will happen. So in this video, we'll learn all these things about action potential in cardiomyocytes. If this video was useful for you, don't forget to subscribe my channel. Thanks for your attention.